So next is the dihybrid cross. It is one of the hybridization cross by considering two characters at a time. Here he is considering two characters at a time. So two parents he selected, one male and also one female. The characters that he considered, they are tall plant with round seeds. At the same time, the female is a dwarf one with wrinkled seeds. These are the phenotypes of male and female. Then how can we write the genotype? Yes, full tallness, capital T, full round, capital R. Like that for dwarf character, small t, and for wrinkled, small r. So next is the step to write the gametes. So while writing the gamete, we want to circle it. So here two letters, two alleles are same. So we can select only one T, capital T only. Here also one R, TR. So what is the gamete? Capital T, capital R. That is the male gamete. Then small t, small r. This is the female gamete. Then production of first generation by crossing this two gametes, male and female gametes. So he got the plants with a genotype capital T, capital R, small r. Capital T, capital R, small r. So what will be the phenotype? What is the physical appearance of this plants? Can you say? Yes, this is tall, round. This is the dominant character, expressed character. That is expressed in first generation. So this female character or dwarf character, a wrinkled character, that is completely suppressed. That is a recessive character. The dominant character is the tall, round character. Then again he conducted one more cross to see, to identify what happened to this dwarf character. Okay, so we can see what is happening in the F2 generation, production of F2 generation. This is the production of, this is the F1 generation. Next is the production of F2 generation by selecting two plants from the F1 generation. Okay, so next we are going to produce the F2 generation, second generation. So how we are producing the F2 generation or second generation? By selecting two plants from the F1 generation, one male and one female. So the male into female. So what is the genotype? What will be the genotype of the male? Capital T, capital R, small r. Then what about the female? The same genotype because two plants are from the F1 generation. Okay. So here the four letters are different. Next we want to write the gametes. The four letters are different here. So how can we write the gametes? Listen here carefully. How can we write? First of all, this T and this T, we want to include one gamete, capital T, capital R, one group of male gamete. Then another one, this capital T with this small r, capital T, small r, second group of male. Then third group of male gametes, that is small t, capital R, this with this two. Then the small t with the small r, small t, small r. Okay, so four groups of male gametes we got. In the same way, here also we'll get this four groups of female gamete. Okay, so it is not easy to cross by doing like this. Okay, so for the convenience of crossing, we can write this male and female gametes in this way. Listen. By drawing columns, we can write the names of male and female gametes. Okay. Here we can write all the male gametes and here we can write all the female gametes. So capital T, capital R, capital T, small t, capital R, small t, small r. The same gametes here also we want to write. These are the male gametes. Capital T, small r, then small t, capital R, then small t, small r. Then crossing. Cross this male gamete with all this female gamete. capital T, capital T, capital R, capital R. Then capital T, capital T, capital R, small r. Then capital T, small t, capital R, capital R. Then this with this one. Capital T, small t, capital R, small r. Like this, next is this gamete need to cross with all this. So in this crossing, we can see now we completed all this columns. Okay. First of all, this male gamete need to cross all with this, then this with all, then this with all, then this male gamete with all this female gamete. So we completed all these rows. Then we want to see what are the characters appeared. So first one is the tall round. Tall round is the dominant character. Listen me. For the appearance of dominant character, only a single gene is needed. If it is a dominant one, capital letters are the dominant ones. So if it is a dominant one, only a single gene is needed. So we can say tall round. How many tall round plants are there? This is tall round. This tall round. 1, 2. This also tall. This is. Sorry, this one is tall round. And this one is tall round. This one is also. These are the gametes. Okay, these are the gametes. This all these are the gametes. This all. Okay, from here we want to count. This is a tall round. This one is tall round. This is also tall round. This also. 1, 2, 3, 4. Then what about this? This here 2 T is there. Then capital R, small r. So this is also tall round. This is no tall round because there is no capital R. So this is tall wrinkled. 1, 2, 3, 4, 5, 6, 7, 8, then 9. How many are there tall round? Tall round 9. The next tall wrinkled. So wrinkled is a recessive character. 2 small letters are needed to appear the recessive character. One is this tall wrinkled. Another 2. Then 3. How many are there? 3 tall wrinkled. The next character dwarf round. Dwarf round. How many dwarf round? So dwarfness is a wrinkled character. 2 small letters are needed to appear this dwarf. That means small t is needed for dwarfness. 1 dwarf round. 2 dwarf round. 3 dwarf round. 3. Then one more character is remaining. What is that? Dwarf wrinkled. That is the female complete recessive character. Only one is the dwarf wrinkled. Okay. So what is the ratio? Dihybrid ratio? 9:3:3:1. 9:3:3:1. That is a Mendel's dihybrid ratio. This ratio is constant when we are taking any other characters other than this tall round. We can select the shape of the flower, color of the flower, shape of the fruit, whatever it may be. What will get? The ratio is always constant. That is 9:3:3:1. This is a Mendel's dihybrid ratio. So based on this dihybrid rules, Mendel suggested 2 more principles. We can see what are the 2 principles based on this Mendel's dihybrid cross. Okay.